So, welcome to the third lecture of week 1 of the course Functional Genomics. So, what we were discussing in the last two lectures are how the discovery made by Mendel led to the emergence of the field called genetics, that led to a new field called genomics, and the post genomics era we are looking at the wealth of the data that is being analyzed using a new branch or rather new approach of science called as bioinformatics. And we said that how it is a workflow, not really a distinct entity. We call it as be it cytogenetics or molecular genetics, and how it has evolved and really help us in understanding. But it is not that bioinformatics is the end of the field that is growing. With all this understanding as to how genes functions and how the genome functions, we are trying to understand something beyond what the DNA sequence tells us. It is not simply the sequence that tell whether you are healthy or unhealthy. It is sometimes beyond your genome sequence. So, that is the field, an emerging field that is coming up, which you call as epigenetics. It is beyond your DNA sequence. So, let us look into what is epigenetics. So, if you look into all of us, let us say that I and my brother, we are twin brothers, we have identical DNA sequence. Is it that both of us will be identical in terms of our physiology, in terms of how our body functions? The answer is not necessarily. The reason being, although we share identical genome, we may not have identical lifestyle. For example, each individual, as it is shown here in this slide, are exposed to different environment, meaning what you eat as a diet could be different. He may be eating something different than me. I may be liking something, for example, burger, he may like pizzas or he may like parathas and so on. I may be a happy person because I am into a job which is stress free, whereas he is into a job which is having much more stress. So, your physiological or psychological state could vary. Or social interactions, I may be living in a country where people are more friendlier, or he has gone to a country where people are not that friendlier, so he is not finding many friends, social interactions. Or I may be tuned to the allopathy, where wherever there is a problem I go to allopathy doctor, which gives you the modern medicine, so to say. Or you may be more a person who likes the traditional medicine and so on. So, the drug that I have, he goes to a different doctor, I go to a different doctor and so on. There are going to be extremely different conditions that we grow up. He may be having a condition which is very cold, I may be in a tropical and so on. So, I may be earning more, he may be earning less. So, that again gives something for you to be happy, or your lifestyle, or how do you take care of yourself. So, I may be not having enough time to play, or he does not like play, whereas he goes and plays. So, his exercise, body activity is different. So, all these things can change the way your genome functions. It is kind of a two-way interaction. Whether you are able to survive an infection is something which your genome has a say in that. At the same time, whether if you are infected, the infection also can in a way change your genome, how does it function. That is what we call as epigenetics. We are going to restrict our discussion to one particular area, that is how this kind of interactions, that is your body interacts with various such changes, whether it is a diet, whether it is exercise, whether it is infection, whether I smoke, whether I drink, what food I have, they have some effect on the genome. And we believe one of the way by which the genome can be altered, but not necessarily at the sequence level, we are identical twins anyway, but it could be that at certain place of our genome, the genome may be modified with a group, for example, which you call as the epigenetic factors. It could be methylation of the histone, methylation of the DNA and so on. So, this changes the way your chromosome functions. You know that when the chromatin or the histone gets methylated, they are more compact. When they get acetylated, they open up. So the expression level of the genes that are present in that region again has a huge influence how the cell responds. So people believe that at the DNA level, for example, the methyl groups that are shown here in this, for example, here this is a methyl group. That is, a methyl group is added to, for example, cytosine, C base of the DNA, can change the way the DNA functions, or it could be, for example, the histone tail, the amino acid sequence that form the histone tail can get, for example, attached with either a acetyl group or methyl group, and then that it affects the way the chromosome gets decondensed or condensed. That dictates whether your gene is functional, less functional, more functional, or not at all functional. So that's one of the ways by which system develops. So your environment or the environment in which you are living, your lifestyle can change the way the gene functions, not necessarily by altering the sequence, but how the chromosome functions, like the modifications. And we know now that these modifications can also have an influence on the next generation. Just to give you some interesting insight into this process, we will talk about what is called as a gut microbiome. What do you mean by gut microbiome? It's very interesting because all of us have microbes living in our body. It could be your gut, the intestine, or it could be your eyelashes, it could be your hair, it could be your skin, it could be your saliva, everywhere. You have microbes thriving, right? And they are, some of them, most of them are friendlier to you. So it doesn't really harm you, but it does, they do have influence on how you function as an organism. So that we will talk about. This is what it is. The human microbiome, only if you will talk about the gut. Our body has 10 times as many microbe cells as many human cells. In fact, if you count the number of cells in your body and the number of microbes that are present, it's almost their DNA is 10 times more than yours. So that is amazing concept. That's what people call it as the second genome. So the microbiome genome is called as the second genome or the other genome. And if you look into that, 90% of your DNA that is there in your body, the microbes have it in terms of the cell number and so on. So that's an interesting concept. If that is the case, that the number of cells that are there in your body of the microbes far exceeds the number of cells of your own, then they certainly are going to have a significant influence on how your body functions, right? So how really this is affected? This is how it is. The gut feeling conflict, meaning there are, your gut has got, this is what just diagrammatically represented, this is your human, the cell of human origin that is there in the body, and this is just to say that in terms of number, they are far above, 10 times as compared to the number of just microbe, that's restricted to your gut. So there is a conflict. The conflict meaning some are good for you, some are not so good for you, right? You don't realize that. But the scientists have realized that the microbiome of your gut has a major influence on your wellness and is important for you to be healthy, and at times it can make you feel sick or fall sick. That's why there are a large number of papers published recently which talks about the importance of understanding the microbes that live in our gut, because it's not same. Even within a family, there are 5 members, each one will have a different types of microbes thriving in their gut, and it changes with age. I'll talk about it a little later. So therefore, the microbiome is going to make a huge impact on your health. So there are of late a number of discoveries have come from the microbiome. They are looking into how it makes you healthy or susceptible for a disease and so on. So that's really something, is because of the genomics revolution. So understanding the importance of the microbes, the NIH, National Institute of Health of the US, which normally steer any new program, has come up with what is called as the human microbiome project, because it is, it's going to be more challenging than sequencing human genome for two reasons. One, it is not unique, because when you talk about the number of cells that are 10 times more than human, the difference is that human cell, the DNA is same in every cell, whereas in the microbiome there could be thousands and thousands of different species that live. So if you take all of them, when you sequence the DNA, it is a kind of a khichdi, you have a mixture. So you don't know which sequence belongs to which species. Even to type and classify them is going to be difficult, because these microbes cannot be cultured outside, because they all can live only inside your body, possibly, right? So even classifying them is going to be difficult. Even governments like Brazilian government has come up with microbiome project. Even the Indian government is trying to put together a consortium or project to understand the gut microbiome Indian population. Remember this, whatever the US government does for its population may not be applicable to India, or even in India every state has a different lifestyle and food habits, right? So you go to Punjab, you will get a food that is very different from the food that you will get from, for example, Tamil Nadu or Maharashtra. So the microbes that thrive or grow in your body depends on what kind of food that you eat. So it's going to be extremely challenging to understand what are the different types of microbes present in the populations, right? But what is interesting is the following: the human microbiota or the biome, it changes. The number of species, the variety changes as you age. This is mostly to do with the kind of food that you eat, right? So that's we can say that the unborn baby is free of any microbiome, because as long as the baby is inside the mother's womb, he or she is completely protected from any microbiome. And when it is born, is free, but he or she gets the microbiome into the intestine when they feed on when the mother breastfeeds. So along with the milk from the skin, the microbes get in, right? There is also a study which suggests that the microbiome of the babies differ between a normally born babies as compared to the babies that are born out of, for example, cesarean section, when they make a small slit in the uterus, take the baby out. So it's different because the kind of exposure they underwent when they are born, right? So that in fact gives some protection or make them susceptible. There are beautiful papers you can go and read. We will post it when the course is online. So you can see that for each, whether a baby, toddler, adult or elderly, this is one study wherein they have shown how, for example, each, you can see that the donut like thing is talks about the different types of microbe. Each color represents a particular group of microbes. So let's not worry about what microbe it is. Let's assume that a color in a donut represents a particular group of microbe, and you can see, for example, this dark green that is seen here in the baby, when in that is almost 50% when breastfed, right? But it goes down, that particular microbiome number goes down if the baby is on formula food, for example, lactogen, Cerelac or whatever it is that when mother cannot feed, then there are supplement that comes. But the moment they shift to solid food, you see that that dark green is disappeared. That means these microbes thrive on the metabolite that come from mother's food or some milk supplement, right? And that change. And then you have another light blue color, whatever, a new group of microbe that come into your gut as you switch to solid food. And of course, you go and you have some fever and your doctor gives you a heavy dose of antibiotic. It's going to wipe out most of the microbe that live in your gut. So you can see that antibiotic treatment led to a majority of them are being this light blue color group. The pink and red has completely disappeared, and you see that a healthy individual who is not under any antibiotic treatment, there is a difference as compared to somebody who is under antibiotic treatment. What is also interesting is that individuals that are malnourished, for example, people or children from marginalized section who are not really having healthy food, nutrient food, their microbiome, gut microbiome is very different. You have this light blue color microbe is almost, the number has come down, and they have this color, this called as the reddish or orange, that group has really outnumbered the other. So that's proteobacteria, right. So we don't know the significance, but what we know is there is a correlation between your profile of gut microbiome as compared to your lifestyle or the food that you intake. And of course, if you are a healthy adult, you have the pink color that is predominant. Obesity, the red again comes down, whether it is malnutrition or you have excessive fat because of you eat a lot, you have the red coming up, right. And as you age, things change. So how do you know this? That there are different groups of microbiome and that changes as you age or the kind of food that you have. We know because of the genomic tools. So basically you isolate the DNA or RNA. Basically here what we do is you isolate the DNA and look at certain signatures in the DNA with which you are able to group the microbes into one or the other groups that what is shown here. For example, firmicutes, actinobacteria and others. So you are able to classify them, and then you are able to classify according to the lifestyle, age and so on. What does it really give you? It's very interesting. This is one study which talks about how your food, the diet, can help or can change the gut microbiome. So for example, fiber gap and the disappearing gut microbiome. This is a paper, this is, you can go and access this paper, it is available. If you give this reference, you will be able to read that. What it says is that the food habit changes the way this microbiome is there. So this is microbial biota, like you see that if you have a good system is something like this, then you have more of fruits and vegetables and less of this junk food. It gives you a microbiota combination which is a very good signature for a healthy living. But if you get into more of this Western diet where more of pizzas and fast food and others, then it tilts, and less of these vegetables and others, it tilts the balance to what is called as dysbiosis, which is probably a pattern which is not in sync with a good healthy life. That is, how does it really help? This is what it is. Your food can really make you healthy or not so healthy. Even your genome otherwise gives you protection against something. So this is the food that you take in, and of course the food is digested, broken, and then all the components are distributed to your body via circulation, and that makes you, for example, your intestinal function to function better, and of course better health, right. But what happens, there could be certain food which you are unable to digest, or there could be components of the food which your body cannot absorb. And that is where it is going to influence what are the bacteria that thrive in your gut. So it is going to change. So if you change the food style, your gut microbiome will change. So this change in the non-digested food or the metabolites or the components is going to really change the microbiome diversity and activity. And if the combination is such that they make lot of metabolites, because they also feed on this food that is available in your stomach, and then they release certain metabolites and they interact with your intestine cells and so on, because they also release many chemicals that would affect or influence your physiology and can make you either healthy, depends on what combination of microbiome you have, or can make you susceptible or put you at risk of developing certain disease. It is not simply anything to do with your digestive tract or digestive disorders. It can affect every part of your body. For example, there are microbiome a particular combination of which is known to increase the heart disease or what is called as inflammatory bowel disease, where digestive process is compromised, you will not be able to sleep, you have stress and so on. Colon cancer, there are studies that have shown clear correlation between your microbiome diversity and susceptibility or risk of having colon cancer. Behavioral disorder, even depression, autism, people are shown to be associated with the kind of microbiome you have. So, it influences obesity, diabetes, asthma, you name it, any of the common disorders that you see are all influenced by the microbes that are present in your gut. Okay? So, you know all these things because of the tool of genomics. You are able to analyze the sequence of the microbes, so you are able to classify them and tell individual A is different from individual B with regard to what kind of microbes that live in his or her gut, and we are coming up with signatures which tell you, okay, this combination is not good. So, you change your food, you change your lifestyle and make a condition wherein your biome is sort of brought back to the normal pattern, therefore, your risk of developing something goes down. So, it is really is translating into medicine, because by typing what kind of biome you have, you are able to tell an individual whether he or she is at risk of developing certain disease. And since the microbiome is dependent on the kind of food that you have and the lifestyle that you live in, you can suggest changes. As a result, your microbiome will change. If that changes, the risk is going to go down. So, it has a translational application, and that is possible only because the genomic technologies that helped in classifying the microbes, typing them, which is otherwise practically impossible using conventional procedures. So, what I spoke about is just one, that is microbiome, but the microbiome after all goes and influence the way the genome functions, which you call as epigenetics, but the epigenetics is not restricted to microbiome. It could be the season, exposure to disease, infection, whether you smoke, drug abuse, financial status, your lifestyle, whether you have exercise, psychological state, social interaction and so on, but these are not independent. For example, you have money or not having money can affect how your brain functions, whether you are happy or not happy. Sometimes if you are a kid, whether you get a food that you wanted or not you didn't get, it's going to affect your brain. Social interaction again, whether you are able to talk to your friend or not in a given day is going to affect your brain, you are happy or not happy. So, things like that, all these are linked, and it is a combination of these that influence your genome as to how they are altered in terms of methylation, acetylation and so on, and that can have profound influence on how you live, and this is something just we are beginning to understand. So, really we do not have all the tools to understand, we are beginning to understand, to see how the change is happening, because we know only one, that is methylation or acetylation. There could be many other modifications that happen in the genome which you have not understood, and how that influence the function of your genes have not been understood. This is something that you need to study. But beyond humans is not, as I told you, the humans are small, less than 1% of all the species that live on the earth. There are many successful species, and that could be of profound use to the humankind. And you can see that if you look into the number of species, bacterium has 4,000 something shown here, protozoans, animals and vertebrates including everything is small, invertebrates enormous number and so on. And what is not included here is the unknown species, which include many of your gut microbiome, skin microbiome, because they cannot be cultured in the lab, it cannot be tested, it cannot be classified. The unknown species mostly in the microbial community is pretty large, right. But if you look into only the known organisms, if you look into the relative number of species that are named, identified, classified, you will find the vertebrates, which includes humans, is pretty small. You have so many other organisms, they are all successful, they live in different conditions, Many of them are of much help to the human welfare, and if you look into that, then it is important to classify, understand the DNA of these sequence, because that is one way you can come up with certain understanding as to how they function, evolve and be successful, right. So, if you see again the prominent biodiversity, meaning the number of plant species and animal species, they are all restricted to certain region of the world. This is world map. The red colors indicate the hot spots where you have a large number of plants and animals that are living. Not all of them have been studied fully, right. So, absolutely these are important, and you can see, for example, India, the Western Ghats from Maharashtra to Kerala and the Sri Lanka, most of these fall in the tropical rainforest, so you have a huge biodiversity, and it is extremely important to understand what the genome of these organisms and how different they are, how similar they are and so on. Therefore, there is a recent phenomena, a new field has evolved, it is called as metagenomics. Herein the goal is to sequence the DNA of a large number of species that are otherwise not studied, could not be studied, cannot be cultured in the lab, because these are not lab organisms. So, you need to understand them, because they have tremendous impact on the human welfare. I will talk about little later. So, this is what they have been proposed and being carried out, is to isolate the microbes or species from different parts. It could be deep down under the ocean, or in the mountain cliffs, or from the seashore, river bird, desert, living organisms are everywhere. Go and collect them, isolate their DNA, sequence them, and look at certain patterns and come up with an understanding. You can go on to do some expression studies, functional analysis, what new genes they have, how that made them to survive there. These are very important, because they have a tremendous application in terms of, for example, agriculture. Maybe certain microbes can tell you the soil texture. So, simply looking at the microbe, you will be able to tell how healthy the soil is, or the microbe in turn gives back something to the soil, right. Or it could be, for example, biotechnology. They may have certain proteins and enzymes which are industrial application, or for example, they could be help us in paper production or leather conditioning, rubber, fiber, you name it. You have huge industrial application. Medicine, for example, diagnosis and others, biosensors, they may simply tell what is the environment that are there. Is it more mercury there, is more iodine there? There are certain microbes that thrive on that. So, it will immediately tell you what it is. Energy, for example, biofuels, food and nutrition. You can talk about everything, and these are the microbes that really going to help us. So, understanding their genome is extremely important. That is why there is a new emphasis on metagenomics, where people are trying to classify bugs or microbes based on the DNA profiling, understand how they evolve, how do they function, and use their knowledge for betterment of the planet, betterment of human welfare. So, with that we will end the third lecture of this week, and we will see you again in the next class.